I will start by thanking the organizers for this invitation. It's really nice to come back to your homeland and tell everybody what you do. So yeah, I work at the MRC Human Genetics Unit at the University of Edinburgh. And one of the missions that we have in the unit is to understand how mutations lead to human diseases. And under this umbrella theme, we have a mission of understanding specifically how sequence changes or mutations in the non-coding regulatory genome, also known as the dark genome, lead to diseases. And a specific category of non-coding regulatory sequences that we focus on is enhancers, which have been introduced very nicely by Pedro. So I would just, for the purposes of my talk, highlight the fact that these sequences are the primary determinants of spatial and temporal regulation of their target genes. And they mainly do so because they are highly enriched in binding sites for tissue-specific transcription factors. And this is true for almost every gene, but it's particularly true for genes that have pleiotropic roles in development. So if you have a gene that needs to be expressed in multiple tissues in multiple stages of development, you can expect it to be sitting in a large regulatory landscape, as Pedro was just showing us, and surrounded by these enhancer sequences. And a subset of enhancers, or sometimes a single enhancer, will control a specific domain of expression of that gene, and this is by recruiting tissue-specific transcription factors to it. So enhancers have been studied for more than 4 decades now, but I think the whole field really came to a boom when we started looking at whole genome sequencing in patient data sets and in genome-wide association studies. And it was realized that a large number of hits that are associated with genome-wide association studies are actually not mapping to the coding regions of the genome. So you can see that in this, I'm missing the pointer, in this pie chart here, that there's a very small proportion of changes that actually map to the coding regions, but a large majority of them map to these non-coding regions. And they are sometimes quite far away from the genes, which is a characteristic features of enhancer. So this big data set that sits there, where we think that these sequence changes are actually sitting in enhancers, and these mutations are affecting the activity of the enhancers, and that's how leading to disease predispositions. And on the other side of the spectrum, we also have a large number of examples where mutations in enhancers have been associated with genetic diseases. So these have been, again, studied for a very long time, and there are cases where there are translocations, so big rearrangements in the genome, where, again, there are no changes in the coding region, but these big rearrangements are messing up the regulatory domains and hence the regulation of the target gene, or they can be deletions, and also quite a few examples where there are single point changes in enhancer sequences that are associated with altered gene expression patterns. So there is, I think, a prediction that there's an order of 1 million of enhancers, and quite a lot of them haven't got characterized activity. So if you like, there's a big challenge here that there's how do we establish functional links between these sequence changes that we observe in these whole genome sequencing data sets and connect them to the etiology of the disease and disease predisposition. And largely the challenge arises from the fact that the way these enhancers have been studied. So as I said, they work by recruiting tissue-specific transcription factors. So understanding what they recruit and what are the specific cell types in which these sequences function in is very important to understand what will go wrong when we have mutations in these sequences. So we have to understand enhancers in the context of where and when they work, and also demonstrate how these mutations are actually altering the expression of the target gene and how that ultimately affects development. So this is the knowledge gap that we are trying to address in the lab. And our aim is to develop methods to visualize and characterize the enhancer active cell population in a context that is relevant to the biology of the disease that we are studying. And in the purpose of this talk, I would be talking about our work on congenital eye abnormalities. And the locus that I would be discussing here is PAX6. So PAX6 is the master regulator of eye development. In any species, losing one functional copy of PAX6 leads to abnormal eye development. And this has been the paradigm locus for studying enhancer-mediated gene regulation for more than 4 decades now. So this is the PAX6 locus. You see the gene here, and all these ellipses that you see around it are the enhancers that have been characterized over several years, mainly by sequence conservation and enhancer reporter assays. And one of the diseases that PRAC6 is primarily associated with is in humans is called aniridia. It's a congenital eye malformation, and patients start off with having a defective iris, and then it leads to progressive blindness. So for a very long time, it was PAC6 sequence was looked at for mutations that are found in aniridia patients, but it was realized that there was a bunch of patients in which there were no coding region mutations, but breakpoints outside the locus which was coding, and these were aniridia breakpoints, and this led to the discovery of these distal regulatory elements. So there are examples of translocations, inversions, and also we, almost a decade ago now, we identified a patient in which there was a single point mutation in this ultra-conserved enhancer, and we could demonstrate, again in the context of a reported transgenic, that there is a loss of feedback regulation. So this enhancer recruits PAC6 amongst other transcription factors. The mutation affects its ability to do that, and this affects the feedback loop and leads to loss of PAC6 expression, thereby leading to an exact same phenotype that you would see by losing a single copy of PAC6 in the coding region. So we've been looking at this locus, and just to give you an idea of the complexity it has, so again, this is the PAC6 locus, and all these ellipses around that you see are all the enhancers that have been characterized, and you can see that the domain spreads around 200 kb on both sides, and all these enhancers together orchestrate this complex expression pattern that is seen, for example, here in zebrafish. So you see PAC6 being expressed in the eye, hindbrain, pineal gland, pancreas, and the neural tube, and we have been able to say which enhancer links to which bit of activity. But this is how we know it at the moment. But all we can say is that this is a retina enhancer, this is a lens enhancer, this is a brain enhancer, but we don't know anything more than that for most of these things. So which is what we want to do now. So as I said, going back to the main question, that we want to develop assays to understand where and when are the enhancers active. And so we want to define the precise cell types, and we want to do it in vivo. So the model of choice here that we have is zebrafish, and I would highlight the important features that are particularly crucial for us. And one of them is that the zebrafish embryonic development is external, and the embryos are transparent, and they develop really quickly. And there are excellent transgenic tools available to generate a large number of transgenic lines that we want to look at. So it's very easy and a very good tool for looking at enhancers in action, which is what we do. So a few years ago now, enhancer reporter assays have been around in the field for a very long time. And the classic way you want to look at an enhancer activity is by hooking it up to a minimal promoter and then looking at the expression of the associated reporter gene. So we evolved that assay a few years ago now, because what we wanted to do was really to evolve it in a way where we can look at two enhancers, for example, the wild type enhancer and enhancer bearing a point mutation, at the same time, so that we can uncover the differences between them. We wanted to define exactly what they do and what goes wrong, but also to compare enhancers that we've been calling eye enhancers, for example, and see if there are specific differences. And we wanted to be able to quantitate those differences, look at the activities of the enhancers at the same time. So the way we evolved the assay was putting the two enhancers we want to look at on the same construct and separating the outputs by strong insulator sequences. So one enhancer drives one reporter, the other drives the other. We can we showed that we can swap these around and the effects are not changed, so there's no reporter bias here. And we also used it for testing known enhancer-associated mutations, for example, wild-type enhancer and the mutant enhancer, and again, flipping them around. And the important thing we did was we created zebrafish transgenic lines where we could dock this construct always in the same integration site, which was bias-free. So the enhancers were not coming under the effect of the enhancers from the native locus. And because they were completely bias-free and on the same sequence, we could quantify the activities of the enhancers and we could co-visualize their activities. So in embryos that are derived from the transgenic lines that bear these constructs, we can look at the expression of GFP and cherry and compare their patterns. So I will not go into details of what we published in that paper, but try and give you an example of what we've been using the system for and how we are taking it forward. So these are so here what we are trying to do is, as I said, we know there are retinal enhancers that we've characterized by looking at them individually in reported transgenic assays. Here, what we are trying to do is compare the two retinal enhancers that we've characterized and see, do they do exactly the same thing or they do the different thing using the two-color system that I just described. So here, the HS5 is driving cherry and NRE is driving GFP. And you can see in these images, if you just focus on the developing eye of the zebrafish, that NRE and HS5 don't exactly have the same expression pattern. There is an overlap in the temporal region of the eye, but it's not like they do exactly the same thing. So there is a spatial difference, and we also investigated if there is a temporal difference. So when Abhishek plays that video, what I want you to focus on is the eye here, and you'll see that the HS5 and NRE are both active in the early stages of development, but as development proceeds, it is mainly the NRE that retains its activity. So we can uncover temporal differences. Yes. So yeah, see that here, they're both active early on, but as development will proceed, you'll see the expression of HS5 would fade away. So it's just highlighting the fact that we can identify differences in spatial terms, but we can also highlight temporal differences. And since we've got these stable transgenic lines on the shelf, they are an excellent source of enhancer active cell populations for us. So what we can do now also is look at these cells in a greater detail and go beyond just calling them retina enhancers and precisely define exactly what cell types they're active in. So we use these transgenic lines as, and fact sort these cells and use them as a source of GFP positive and cherry positive cells. And then when we look at this particular line, you can see we can identify a lot of retinal cell populations by doing single cell RNA sequencing. And what is important is that we can go in and look at that data and then identify exactly what cell types NRE is active in. So for example, in this case, based on GFP expression, NRE-driven expression is seen in retinal stem cells and in amacrine cells of the retina. Cherry-driven, HS5-driven cherry expression is seen in neural glia, retinal progenitor, and retinal stem cells. So there are cell types where they're both active, and there are cell types that they're uniquely active. So just going back to the point that we want to do, so where and when are these enhancers active? We've now have got methods to go beyond just calling them eye enhancers or retina enhancers. We can precisely define the cell types, and we've been using this to compare the wild type and mutant enhancers doing exactly the same assays. So, remember the mission. The question is that good enough? So, I can tell you where enhancer is active, exactly what cell types it's active, how the mutation is affecting that activity. But I haven't got the power in the zebrafish system to delete the enhancer and then demonstrate that this cell type has gone lost and the expression or the development of that organ has changed. And this is simply because zebrafish underwent a whole genome duplication event, and there are duplicated copies of PAC6, so it's not a suitable system. So if we want to go to from when and where to how, we had to develop another system in the lab, which is, again, we wanted to keep it relatively high throughput so that we can look at many enhancers at the same time. So what we've been looking at in the context of eye development, establishing this optic cup organoid differentiation system, where we have mouse embryonic stem cells in which one of the genes that comes early on in when the embryonic stem cells start going towards making eye-like structures, has been tagged with GFP. So we got these cells. And when we culture them in specialized dishes, in specialized media, these cells spontaneously differentiate around day 6 to form these optic cup-like structures. We don't take them beyond 7 days, but you can take this culture up to 28 days and get a fully formed retina. So we wanted to see if we can model loss of changes in PAC6 expression by mucking up with the enhancers that we have been working on. So in other words, can we use this system to test the hypothesis that we generate in the zebrafish? So what we did was, again, going back to this enhancer that I was talking about in my introductory slides, where we found a point mutation that was identified from an aniridia patient, which is shown here, that has the exact same phenotype as losing a single copy of PAC6. So what we decided to do was delete this enhancer from the embryonic stem cells I was just talking about and see if that affects the development of the optic cups. And so we had this model, but it was all generated in the context of a reported transgenic animal. We wanted to test that model in the context of in an ex vivo differentiation system. So this is what we did. And you can see in these clones that when we take out 2 copies of the ultra-conserved enhancer, which is called SIMO, in 3 independent clones, we see that the size of the organoids is massively shrunk and they do not form optic cups anymore. So there is a failure of differentiation defect and the optic cups are no longer formed. There is a phenotype in the heterozygous deletion as well, but it isn't as stark as what you see by losing both copies of the enhancer. So there is a clear differentiation failure of these cells towards optic cup-like structures. And then when we go in and do RNA sequencing on these clones, this is just confirmed by we see a downregulation of genes that are important for eye differentiation. So these cells have been arrested in differentiation state, and we can clearly see that the homozygous deletion clones cluster away from the wild type and the heterozygous. There's a small phenotype that is visible here as well. So we wanted to look at what happens to PAX6 expression. So we've been doing some immunohistochemistry on these clones. So first of all, what we noticed, just using the wild type optic cups, is that the expression of PAX6 actually comes on earlier than RAX expression, so which is seen here. And the number of double positive cells markedly increase as we take them to the next steps of differentiation. So by day 7, there is a significant increase in the number of double positive cells. So PAX6 comes on, and then the GFP expression comes on as well. And so there's a trend that is an increase of double positives that we see in the wild type, where we see an exact opposite trend, loss of PAX6 expression, and then the number of double positive cells decline sharply. So what the stage that we are at, so again, going back to the diagram that I had in the beginning, so we had this mutation in a transgenic model. We could demonstrate that this effect on PAX6 expression, but now we can actually show that deleting this enhancer affects PAX6 expression and affects eye development in eyes in a dish, if you like. So this gives us a window into deleting more PAX6 enhancers and then looking at their effects, and then maybe going to individual cell type specific resolutions in the system as well. So, but is that good enough? So, what I have shown you so far is a story about 3 PAX6 enhancers, which is like 8 to 10 years of work by 2 PhD students and a technician. And we've been able to answer some questions about these 3 enhancers, but we've got a lot to cover here, and this is just one locus. So what we thought is that we should be looking at increasing throughput, but also looking at ways of asking more complex questions, like major rearrangements in the locus, taking out all the eye enhancers together, things like that. So we've been integrating synthetic biology approaches into our pipeline. So use synthetic biology to make these constructs artificially, and then that will give us the power to make many, many more rearrangements, deletions, docking in mutations, and then again, using the same systems that I've been talking about, in vivo imaging, single-cell RNA sequencing, to kind of characterize the changes that we've been making. So this is where we have a collaboration with Professor Jeff Buki in New York, and what we've been looking at is synthesizing these loci using synthetic biology. So again, as I said, we have a number of rearrangements that we've been making, deleting enhancers, sequence changing, introducing sequence changes that are associated with diseases, and rearrangements of the regulatory sequence, and we've been testing them in the fish models. So what we've been doing recently, so while this huge 700 kb locus is being synthesized and delivered to us, we cheated using evolution a little bit. So PAX6 is highly conserved, and in this fugu fish, actually the whole locus still retains the enhancers, but it has been compressed from 700 kb to 70 kb, which was well within our realms of synthesis and integration to start with, just to test the system and get the pipelines running. So Jeff's lab synthesized this artificial construct for us, which has all the enhancers and the PAX6 gene, and then integrated 2 different reporters. And we put this massive 70 kb construct in our zebrafish transgen and generated zebrafish transgenics. And as you can see, it recapitulates the complete expression pattern of PAX6 in both colors. So there's a lot to do here, and we have a number of constructs that we have been given that we are analyzing at the moment. So what we are trying to do is humanize the locus so that we can still work with Fugu, but we have human enhancers in place, make this compatible so that we can start using the optic cup differentiation system, and also do big changes and ask some like basic questions about enhancer biology that are really outstanding in the field. So yeah, this is just my summary slide and maybe the take home that, what we are trying really to do is to develop ways where we can visualize and characterize enhancer active populations. The context is really important. So we try and stick to that. And we are trying to go from low throughput, moderate, and hopefully one day to high throughput analysis. And the aim really is the mission that we have in enhancing the understanding of the underlying disease etiology, understanding these mutations and how they lead to diseases. So yeah, I'll stop here, take any questions, and thank all the funders. And yeah, if you fancy another meeting, want to find out more about rare cell populations, dark genome, you come to Edinburgh. We are organizing this in spring next year. Thank you. Happy to take any questions.
Thank you so much, Shipra. Questions?
Hey, thanks. This is really very nice. Just for my own understanding, there's a lot of correlations between the looping, enhancer looping, and your, the retinal defects, right? How do you think of establishing a causal link? Is there a way of CRISPR insertion, gain of function?
We can do that, and we've been trying to knock the mutation that I was talking about. But I think the problem we've always had is like, we know where to look, and now we know where to look, so we can definitely engineer these changes and then look at the altered function of the precise cell types where these enhancers are active in, which is where we are going. So yeah, I mean, the approach would still be CRISPRing in, or maybe the synthetic biology would kick in and we don't have to CRISPR things anymore. So, because we have the whole locus there, so we are not looking at a standalone enhancer. We are looking at the whole locus and then the change and so it'll still be reporter gene expression, but at least we will have the full context and not just one enhancer in picture.
So in the in vitro model you had, what is the time scales of the manifestation of the function?
The organoid differentiation system. So what we have been, the enhancer that we're looking at, we see defects from day 5. But we haven't taken the system, so it would depend. Some of the enhancers are late acting, that they act in the late retina. So maybe there we will have to look at day 10 or beyond. And there's not that much information available that we can precisely correlate that to mouse embryonic development. So again, that's a gap that we need to fill.
Okay. Yeah, Shipra, I had a question. So given that PAX6 heads have a phenotype, do you think haploinsufficiency or pleiotropy of PAX6 expression itself is also important to look at? So how do you think your enhancer elements could affect that? And is there a way to study a gene dosage effect in your zebrafish mutant models?
In the fish, no, because we are not even looking at PAX6 there. We take the sequence out, we put it somewhere, and it does something. So yeah, no. But in the organoids, yes, because as I showed that taking one copy out also has an effect, which is not significant. So it's not going to be as drastic as taking a single copy of PAX6 out, and it'll be in very specific cell types. But yeah, we can model that when we are looking at PAX6.
Okay, yeah, thank you. If there are no more questions, let's thank Shipra. I think we're going to break now for coffee, and then we'll come back at 4 and resume the session.